Good morning, Doc. Good morning, everyone. As the representative of Block D, Group number 2, to discuss about task number 3, which is to tabulate 5 elements involved in gene regulation and 1 corresponding application for each. We have enumerated the following 5 elements. These are core promoter, enhancer, silencers, insulators, and proximal promoter. Now we have to take each one of it to discuss and give at least one application for. First, we have the core promoter. It is a portion of the proximal promoter that contains transcription start sites. It serves as a docking site for the transcriptional machinery and pre-initiation complex assembly. It contains a binding site for RNA polymerase. The vast network of regulatory factors that contribute to the initiation of transcription of RNA polymerase ultimately target any specific gene's core promoter. Next, we have the enhancers. It contains binding sites for the same transcriptional activators, and they basically enhance the gene expression. Enhancers is a short region of DNA that can be bound by proteins to increase the likelihood of transcription of a particular gene to occur. We have the silencers. Its effects is opposite to enhancers on the transcription of the target gene and results in decreasing their level of transcription. It contains binding sites of repressive transcription factors. A DNA sequence capable of binding transcription regulation factors called repressors. When a repressor protein binds to silencer region of DNA, it prevents the transcribing of DNA sequence into the RNA. With transcription blocked, the translation of RNA into proteins is impossible. Thus, silencers prevent genes from being expressed as proteins. We have the insulators. Insulator region limits the effects of other regulatory elements to certain chromosomal boundaries. They create regulatory domains unstained by regulatory elements in region outside the domain. Insulators can block enhancer-promoter communication and/or prevent spreading of repressive chromatin domains. Insulators function either as an enhancer or blocker or a barrier, or both. The mechanism by which an insulator performs these two functions include loop formation and nucleosome modification. The genetic implication of insulators lie in the involvement of mechanism of imprinting their ability to regulate transcription. Mutations to insulators are linked to cancer as a result of cell cycle dysregulation and silencing of growth suppressors. And lastly, we have proximal promoter. Typically right upstream of the core promoters and usually contain binding sites for activator transcription. They provide additional control over gene expression. And for a quick recap, we have tabulated the 5 elements involved in gene regulation and its corresponding application. And a video clip will follow right after. Thank you for listening.
Hello? Are we done? Hello, Christian?
Dok, walay kuan dok audio ang video gi tarong pa dok.
Okay, sige, sige daw.
Thank you.
Okay, so to add na lang, no, it's not only those, no, so, okay man po to, what I want you to answer, aside from the things that we mentioned, these are, yeah, mga non-coding genes na po na siya, which are used in genetic regulation, in what I mean is katong mga enhancers, promoters, so these are still DNA, RNA materials that are not involved in genetic in protein protein synthesis, no, but mana siya, these are used in mga transcription, mga no, or even translation. But aside from that, you also have, do not forget, no, uh, the other non-coding DNAs such as the SNPs, no? As we mentioned, disease susceptibility. Second is the copy number variations, okay, which has almost the same functions, but ang SNPs is, these are more significant. Next is the microRNA, no, which is basically the function of microRNA is for post-translational silencing. So post-translational, there's already translation as transcriptional. Let's check. Post-transcriptional silencing. So there is formation already of RNA, but RNA is not being stopped to being formed to protein, okay, in order to silence, no? So what's the purpose of silencing this translation or post-transcriptional formation of conversion of RNA to protein? It's to prevent the formation of disease, diba? So post-transcriptional, again, correction na post-transcriptional silencing, that is microRNA. For the what's the other one? Not long non-coding RNA. It has, aside from silencing, it can also have other functions, activation, linking with other complexes. It's there in the figure. And do not forget also the epigenome. Epigenome, these are materials which are not DNA. It's above the DNA level. We are talking about the histones. We're talking about the histone modifying factors, no? If you know, if you were able to read DNA methylation, which is involved in transcriptional silencing. So it's different from post-transcriptional silencing, wherein you already have a RNA formed. But for DNA methylation, the formation of RNA is being stopped. RNA mismo walay na form na RNA. So that is DNA methylation. So how is this important? Again, it is applied in treatment of cancer, no? In the treatment of cancer, especially when the cancer is linked to any bang, for example, the problem is a problem in the gene, histone, I'm sorry, deacetylase, no? So if it's a problem with the enzyme, then what the remedy is to treat the patient with that enzyme, diba? Or an inhibitor of that enzyme. Okay, so that mga po ng application sa epigenome. Then you can also mention, okay, you mentioned about cancer diseases, also mention about personalized medicine. Okay, so that means we might have, for example, two patients might have breast cancers, the same diagnosis, but looking at it, what the genes involved are different. So different food ang treatment. And okay, so ana ang application. So let's continue. So nag-segue lang tagamay, no? Maybe you so let's continue with the report of the third group. Thank you.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Stop. That ends our reporting for
sorry.
group freedom.
Hindi kayo multitasking mo ko diri. Sige. Okay, so thank you so much, no? So next group. So let's go to the group question number 4, group number 2 of section B.
Good morning, Doc. Good morning, classmates. So we are group 2, and I'll be representing group 2, and we'll talk about the Human Genome Project. So the Human Genome Project was done in the 1990 to 2000 1993 until the recent. And then its aim and objective is to sequence the whole genome with 3 billion basis points. So also its aim is to create a physical map of a human genome, and then to identify all the disease-causing genes. And then also it enables us to understand the function of the genes, then make the informations available for the future researchers, and then to develop tools for processing and analyzing data. So I will be showing a video. This talks about the history of the
Packed inside
whole
every
genome
cell in your
project.
body is a set of genetic instructions, 3.2 billion base pairs long. Deciphering these directions would be a monumental task, but could offer unprecedented insight about the human body. In 1990, a consortium of 20 international research centers embarked on the world's largest biological collaboration to accomplish this mission. The Human Genome Project proposed to sequence the entire human genome over 15 years with $3 billion of public funds. Then, 7 years before its scheduled completion, a private company called Celera announced that they could accomplish the same goal in just 3 years and at a fraction of the cost. The two camps discussed a joint venture, but talks quickly fell apart as disagreements arose over legal.
Nanay, but home service. What?
All the difference. Their paths diverged in the most critical of steps. That overlapped each other a little bit on both ends. Each of these fragments of DNA was inserted inside a bacterial artificial chromosome where they were cloned and fingerprinted. The fingerprints showed scientists where the fragments overlapped without knowing the actual sequence. Using the overlapping bits as a guide, the researchers marked each fragment's place in the genome to create a contiguous map, a process that took about 6 years. The cloned fragments were sequenced in labs around genome. The consortium's better safe than sorry approach contrasted starkly with Celera's strategy, called whole genome shotgun sequencing. It hinged on skipping the mapping phase entirely, a faster though foolhardy approach according to some. The entire genome was directly chopped up into a giant heap of small overlapping bits. Once these bits were sequenced via the Sanger method, Celera would take the formidable risk of reconstructing the genome using just the overlaps. But perhaps their decision wasn't such a gamble, because guess whose freshly completed map was available online for free? The Human Genome Consortium, in accordance with the project's second major principle, which held that all of the project's data would be shared publicly within 24 hours of collection. So in 1998, scientists around the world were furiously sequencing lines of genetic code using the tried and true yet laborious Sanger method. Finally, after 3 exhausting years of continuous sequencing and assembling, the verdict was in. In February 2001, both groups simultaneously published working drafts of more than 90% of the human genome, several years ahead of the consortium's schedule. The race ended in a tie. The Human Genome Project's practice of immediately sharing its data was an unusual one. It is more typical for scientists to closely guard their data until they are able to analyze it and publish their conclusions. Instead, the Human Genome Project accelerated the pace of research and created an international collaboration on an unprecedented scale. Since then, Robust investment in both the public and private sector has led to the identification of many disease-related genes and remarkable advances in sequencing technology. Today, a person's genome can be sequenced in just a few days. However, reading the genome is only the first step. We're a long way away from understanding what most of our genes do and how they are controlled. Those are some of the challenges for the next generation of ambitious research initiatives.
So that was the history of the human genome. So now, let me talk about the medical application of the Human Genome Project. So this is just additional information, but this is not the whole thing because there's a lot of medical application for this, and I will only cite 3. So medicine will be able to look, in medicine, we will be able to look more into the fundamental causes of diseases rather than just concentrating on the treating symptoms. So basically, before, they were only treating symptoms because they really didn't know the main reason for one person's disease. So manifestations were visible, but the real cause weren't. So at the current, aside from treating, it can also be prevented through genetic mapping. And then next, genetic screening, we will be enable rapid and specific diagnostic tests, making it possible to treat countless maladies or diseases. And then the DNA-based test clarifies diagnosis quickly and enable geneticists to detect carriers within the family. So this statements will come out in the hereditary diseases. So it can be prevented if we will be able to imply it in the very first place. So just like cancer, if ever you have a family or history with breast cancer, so you might just have to remove or mastectomy or do some like, for example, some artists remove their breast because they have predicted that their family will pass 85% of those cancer genes. So that's it. And then recall the recent research findings regarding the human genome. So first, we have genetic variations or polymorphism associated with diseases located in our non-protein coding regions of the genome. So majority of diseases associated with genetic variants are located outside of protein coding regions. So these regions are other than the DNA sequence. And then they affect regions that control transcription, such as promoters or enhancers, and then non-coding RNAs that influence the expression. So one research was about the isolated Pierre-Robin sequence. So this caused by disruption or damage of enhancers that regulate the activity of SOX9 gene, a gene that provides instruction for making protein that plays critical role in embryonic development. So what does SOX9 means? So this is a gene activity in which it is reduced that it cannot properly control the gene essential for normal jaw development. So this will cause micronathia and consequently glossotosis, then airway obstruction, and in some cases, this will cause cleft palate. And then next, any two humans are typically greater than 99.5% DNA identical. So it only varies in the variation encoded in the less than 0.5% of our DNA, or the 15 million base pairs. And So it is a genetic, we can say that it can be a genetic mistake or that can determine our differences. So in this, we can some will cause diseases and some will cause the manifestation of our color or the tallness or the shortness of a person. And then in addition to this also in a differential susceptibility to disease. So in genetic variations, there can also be a large or small effect on the likelihood to developing a particular disease. For example, breast cancer or ovarian cancer. So a person may have developed or may not. It depends on the gene expressed in them and the percentage that it has or the mutation occurred. So also environmental exposure. So the expression of genes in an organism can be influenced by the environment, including the external world in which the organism is located or developed. So a research from Jackson's laboratory, they studied the mice. So these mice in the picture, they have the same mother, but they have different color. So how can two mice with the same DNA sequence have different coat colors? So it is due to epigenetics. So in this particular yellow mouse strain, when a mother is given a diet enriched in folic acid, a good source for epigenetic, which is a good source for epigenetic methylation of DNA, this will control the coat color, which is in methylated DNA, it will result into more brown pups being born than yellow. So this is an example of how a diet can cause epigenetic changes to affect an individual without physically changing their DNA. And then next is we have epigenetics. So what is epigenetics? So it is a study of heritable changes caused by the activation and deactivation of genes without any change in the underlying DNA sequence of the organism. So this happens when the DNA form in the histone. So so basically it is the relative state of the DNA and then the histone acetylation or methylation. So if this happens, histone residues tend to condense the DNA and lead to gene silencing. So if there is a methylation, even in DNA itself, a modification that is associated with transcriptional inactivation will occur. So also in the non-coding RNA, so we have two microRNA and the long non-coding RNAs. So microRNA will play a role for biochemical markers, like for example, the mRNA in cancer. So mRNA or microRNAs are large group of small non-coding RNAs that control post-transcriptional expression of target genes. So in this, the post-transcriptional silencing, it will produce a primary RNA. So that's the function of the non-coding microRNA. So if there is a mutation that will occur, it can cause a cancer. And then for primary important role for cancer also includes long non-coding RNA. So basically this happens in the PV1, which is studied by substantial evidence lang ni siya, guys, indicates that this plays a critical role in the onset and development of cancers because PVT1 acts an oncogenetic factor by promoting cancer cell proliferation, invasion, metastasis, and drug resistance. Just like a microRNA plays a role in cancer because if there is a suppression of cells and then the changes will occur, there will be proliferation of cells which will cause cancer or become a cancer cell. So that's it. Thank you, guys. That's
Okay.
it for my report.
Thank you so much, Sarah.
Thank
Okay.
you, Doc.
Sige. So, okay, well explained, no? So, mana siya on the applications. We're talking about applications of these. So, to add, no, epigenetics does not only tell us about at the molecular level, but simply speaking, epigenetics tell us about the outside factors, no? For example, there's this one example about twins, no? Siblings na twins ba? They are genetically the same. I'm talking about identical twins. This genetically the same, pero why will there be differences? No, of course, differences because of difference in experiences, stressors, in the, for example, lahi man gid pud na, no, in the upbringing, no. So these are epigenetics, also included in the epigenetics. So not only in the molecular level, but we can also cite examples on the physical, in the clinical level. Mga drugs taken by this person, no, by this twin, not taken by this twin. So ano siya, epigenetics. So anything that is outside, now this is important, no? Epigenetics is very important because all the while before, in the old school of thought of diseases, diba? We thought before that diseases arise due to a defect or a problem, mutation in the DNA. But now it's not anymore like that. No, it's aside. It can also be DNA, no, be DNA based, but you can also have this epigenetics. All other factors outside the DNA. So pwede protein, histones, anything. Stress, medicines, chemicals, radiation, these are epigenetics. So ang analogy ana is nurture and nature. No, nature is the DNA, nurture is the epigenetics. Pwede po na culture, no? Culture, example, how one person perceives things is affected by the culture of the family. Epigenetics po na siya. Okay, so what's next? What's the number for group 1 of section D? Thank you.
So good morning, everyone. I'm Dana Tibay, and I will be reporting in behalf of Group 1 from Section D. And our next slide, please. So for instruction number 5, discuss the role of epigenetics and cite examples. So epigenetics is the study of heritable chemical alteration of DNA or chromatin that can activate and deactivate genes without changing the DNA sequence. So in other words, epigenetics, it refers to the process that allows an individual's gene expression to be altered without altering the sequence of their genome or their DNA. So epigenetic modifications have the ability to activate and deactivate genes as well as determine which proteins are to be transcribed. So there are three systems that can interact with each other to silence genes. Number one is DNA methylation. DNA methylation refers to the addition of methyl group to the DNA strand itself, where it typically results in transcriptional silencing, and it is tightly regulated by transferases, demethyling enzymes, and methylated DNA binding proteins. Next is the histone modifications. Like histone methylation, histone methylation is the modification of certain amino acids in histone protein by addition of one, two, or three methyl groups that results to silencing. And for the histone acetylation, um, lysine residues are acetylated by histone acetyltransferases, and it results to activation. But changes can be reversed by histone deacetylases, also known as HDAC. And for the histone phosphorylation, it attaches to serine of histone and results to either activation or inactivation. And last is the RNA-associated silencing, like the microRNA and long non-coding RNAs discussed by Group 2 earlier. And this epigenome also has lifelong dialogue between genes and environment. So this is where the katong nature versus nurturing that was mentioned by Doc earlier, and it can be influenced by factors including the diet, chemical exposure, and medication. Environmental epigenetic changes are the very reason why genetically identical twins can grow up to have a very different lives. Next slide, please. So, in the research that we found, social experiences can also cause epigenetic changes. The phenomenon of nurturing, studied by Weaver et al., stated that some rat parents who licked and combed their offspring intensively grew up to be more happier and peaceful, whereas those who are ignored grew up to be anxious and disease prone. So for our epigenetic example, we found, next slide please. We found this fascinating study about the epigenetic markers between the Oromo and Amhara peoples of Ethiopian highlands. So Ethiopian highlands is a place in Northeast Africa which has a 4,550 summit elevation. So the researchers found out that the Oromo peoples who were the migrants of the place, they were expected to have an increase in epigenetic marks around genes associated with oxygen uptake or red blood cell production, because these adaptations in the human body allowed the Amhara peoples, who are the native of the place, to allowed them to live successfully in their elevated homeland. But instead, the researchers found that the increase of epigenetic markers were around the genes associated with the immune system, and this appears to show that epigenetic marks acted as catalyst for the introduction of variation in gene expression, and it allowed the people of Oromo to have the defenses in their body to combat the new microbiological threats that the migrating population were exposed to upon arriving to the highlands. To explain further, we have a video. Krish.
Susan and Sharon are identical twins who have the same DNA. Genetic information stored in the cells in a certain order or sequence that serves as a code to tell their bodies how to function. DNA determines what the genes do, the chemicals they produce, and what health risks they're predisposed to throughout their lives. Even though Susan and Sharon have the same genetic information, they're not alike in their personalities and behaviors, or in their health as they age. Susan gains weight and has trouble losing it, while Sharon maintains a healthy weight throughout her life. Susan develops cardiovascular disease, while Sharon doesn't. If they have the same genes, how can that happen? Clearly, DNA is not our destiny. New evidence shows that we can change the effect of our genetic makeup on our health. For example, when pregnant, women's experiences, including behavior, diet, and environment, can affect the gene expression of their child without changing the child's DNA sequence. The influence of external factors on our genes is called epigenetics. In addition to influencing which of our genes express themselves and how, epigenetic mechanisms may play an important role in the development, diagnosis, and treatment of diseases. By understanding and influencing epigenetic mechanisms, we can optimally experience the 4 crucial components of our gene expressions for a healthy life. Potential are gene risk factors and influences. Capacity, external effects on metabolic processes and our health status. Balance, through critical nutrition management and behavior change, we can maintain optimal methylation for our genes to be most favorable for health. Expression, our optimally managed health status. There are 2 important epigenetic mechanisms: DNA methylation and histone modification. The first is histone modification. In all of our cells, our DNA is wrapped around proteins called histones. When the DNA is wrapped tightly around the histones, the genes are unreadable and they are not expressed. When the DNA strand is loosened around the histones, genes are accessible and may be read and expressed. In this way, modification of histone proteins can turn a gene's expression on or off. DNA methylation occurs when chemical switches called methyl groups are added to or removed from regions of the DNA. Methyl groups affect the expression of the genes in that region of DNA by turning them on or off. These 2 epigenetic mechanisms can explain why Sharon and Susan, though they have identical genes, have different health outcomes. Their different environmental exposures or their health behaviors may have led to different epigenetic influences, and therefore to their different traits and health outcomes. Epigenetic mechanisms like histone modification and DNA methylation are complex. The DNA methylation process involves several metabolic gears that produce the necessary methyl groups for the methylation process. Each component or gear in the methylation cycle is important for the process to continue, and several nutrients are required, including vitamin B12, vitamin B6, and folic acid. When there are non-optimal levels of folic acid in the body, the gears of the methylation cycle don't work properly. When they move too slowly, the methylation process is disrupted. Different diseases may develop, including cancer, cardiovascular disease, Alzheimer's disease, and diabetes. One example of when the gears of the methylation cycle could slow down is when a person has a variation of the gene MTHFR. A variation, or polymorphism of this gene, causes metabolism of folate to be changed. Variations in MTHFR are common in the population and may affect up to 40% of us. People with MTHFR polymorphisms have increased risk for several diseases, including cardiovascular disease and some cancers. Outcomes that can be influenced by genetic polymorphisms especially affect people who have 2 copies of the polymorphism in their genetic code, also known as homozygous expression. Having 2 copies of the genetic polymorphism means that both parents passed on the gene polymorphism to your genes. Being heterozygous, having just 1 copy of the polymorphism, means the risk of disrupted methylation is reduced but still present. Identifying genetic variations and our methylation capacity may be done by simple assays of blood. Blood is easy to access and can reflect metabolic and inflammatory pathways and outcomes in our cells, including levels of important blood chemicals like homocysteine, S-adenosylmethionine, and S-adenosylhomocysteine. Identifying methylation capacity and balance in blood chemicals with these assays allows healthcare providers to identify high-risk patients like Susan, who may benefit from health interventions. Nutritional interventions and therapies like adding folate to the diet target and may improve the methylation process. Unlike mutations and other genetic abnormalities, epigenetic modifications for our gene expression may be changed by changing our behavior. The study of epigenetic markers and mechanisms is emerging as one of the most promising strategies for identifying and treating patients who have or may develop complex diseases. When genetic testing and related therapies are implemented, we will be able to improve public health.
That would be all, Doc. Thank you.
Okay, thank you so much. So, which is the last group? Last group na tanong? That will be group 3 of section B.
So hello, everyone. Good morning. My name is hello. Am I audible? I'm sorry. Hello.
Mabatian ra ka.
Okay, thank you, Doc. Bye. Okay, so good morning, everyone, and future doctors. Good morning, Doc Lau. So my name is Mary Ann Sarahena. I'm from Section B, Group 3,
Morning,
and we're
Mary.
assigned to discuss about gene editing. Next slide, please. Okay, so here are my members with our leader, Steve Angelo. Next, Doc. Okay, so before we discuss the main topic of our session, let's just have a refresh of what is a gene. So it's a basic unit of heredity that is passed from parents to offspring that contain the information needed to specify the traits. It can be by the color of the eye, the height, color of the hair, and many more. A segment of DNA needed to contribute to a function. A set of genes that's called genome are being received that sets as an instructions and shuffles throughout the generation. But there are actually faulty genes or errors that leads to mutations and diseases. That's why gene editing has been developed. Next slide, please. Okay, so gene editing is a group of technologies that give scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at particular locations in the genome. There are several approaches to genome editing that have been developed. A recent one known is the CRISPR-Cas9, which stands for clustered regularly interspaced short palindromic repeats, and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate, and more efficient than other existing genome editing methods. So next slide, please. So let us discuss what is a CRISPR. So it stands for clustered regularly interspaced short palindromic repeats. It is a family of DNA sequences found in the genomes of prokaryotic organisms such as bacteria and archaea. These sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryotes. So CRISPR-Cas9 was adopted from a naturally occurring genome editing system in bacteria. The bacteria captures snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR arrays allow the bacteria to remember the viruses. If the viruses attack again, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then use Cas9 or a similar enzyme to cut the DNA apart, which disable the viruses. Next slide, please. So CRISPR is a genetic engineering tool that uses a CRISPR sequence of DNA and its associated protein Cas9 to edit the base pair of a gene. So if you can alter the DNA of an organism, you can basically customize most of its genetic features simply by repairing or replacing a gene. So in the best case scenario, CRISPR could be used to solve cellular anomaly disorder like cancer, Alzheimer's, blindness, sickle cell, and many more. and it allows scientists or researchers to easily alter DNA sequences and modify gene function. I understand and I know that these terms are very new to us, most especially the CRISPR-Cas9. So at this moment, let's have a grasp of understanding regarding gene editing and how the CRISPR edits the DNA. So let's watch the video.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors, like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics, or check out some of the other mind-blowing science being developed around the world and in the universe right now.
Okay, so I hope we had a grasp or a little of understanding of how CRISPR edits the DNA and what is gene editing in general all about. So to summarize the topic, the DNA contained our genes like an instruction material for our cells. Gene editing is applicable when there are faulty and error genes that need to be replaced, since these genes causes diseases. So CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases, and improving the crops. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. And also, there are some ethical and societal concerns to consider. So, at the next slide, we would see the positive. First is the positive contribution of gene editing. Next slide, please. Okay, so for the positive contribution, this is to be able to search for new therapies or treatment from certain diseases such as cancer, leukemia, HIV, and can edit genes that cause hereditary diseases. Also, editing the genes of certain vectors like mosquitoes to stop spreading malaria. Next slide, please. And this time we have the downside of gene editing. They could actually invent designer babies. As far as I've read about it, it's more of like modifying features, or we can select features of the babies that we want. Like for example, we want them to have a green eye or a very sharp nose, and then it's like a click of our fingers, we could like order them and like we have designer babies already. And also we have it could actually develop more virulent microbial diseases if it's used on the negative side. So now modern technology is in our hands. Now it's time to debate how we use it, regulate it, avoid negative uses, and unlock its potential to provide life-changing solutions to some of the world's biggest challenges. So next slide, please. Okay, so let us end the report by a quote. Develop a passion for learning. If you do, you will never cease to grow. This is by Anthony D'Angelo. That would be all, Doc, and I hope you all gain a lot of learnings from this report. Thank you, and God bless.
Thank you so much, Mayor. So there. For the exam nga tong andaman ani no. Okay, for the exam next week, okay, be able to know. I'm very particular with the figures in the book. No, what I mean is the drawings, the processes. Be able to study that figure in gene editing, no, mga Cas9 protein, etc. What is the concept involved in what they call this in post-transcriptional silencing, no, and microRNA, okay, as well as the long non-coding RNA, okay. So mana siya kanang mga concepts, no. So I'm very particular because this is only a short lesson, no. 6 pages lang siya sa book. So mana kanang sa paman. What else? Any questions about this topic? Klaro ra? So, any questions, class?
So far sa section D, Doc, wala ra.
Wala ra. Section D? Section D?
So far, Doc, wala ra sa Doc.
Wala ra pod. Okay. So ana lang review lang gyud on the I'm very particular what is the application? What is the best reason for considering epigenetics, mga na ba, mga application ako mga questions, very easy questions. I also have some like 5 cases, no, few cases pa karon, more on conceptual ni siya akong example. Okay, class? Okay, sige. So let's call it a day and have your lunch now, or if you have an exam, you can study in advance. Okay, so the exam next week will be on Wednesday, 7 to 8 a.m. So it will be until strictly 8 a.m., but I will open the exam as early as 6:45 a.m. Okay?
Thank you, Doc. Noted,
Okay,
Doc.
thank you so much.
Thank you, Doc.
Thank you, Doc.
Okay, take
Thank
care.
you, Doc.
Okay.
Thank you,
Bye.
Doc.
Thank you, Doc. Thank you, Doc.
Ah.